Hi everybody. This week we are going to study target identification and mass spectrometry. Again, I chose several references from Wikipedia. And in this lecture, we are going to study, first of all, what is target identification? And there are many different kinds of target ID technique, and we are going to compare those differences among them. After that, target validation, and we will learn what is a target VD and its meaning. Last part will be mass spectrometry and its application to chemical biology. Ready to go? Okay, in fold chemical genetics, we said we start with the phenotypic screening, and then we give shock with the chemical tools. So this small molecule can go inside the body and induce some kind of phenotypic changes. And then next step is we need to understand the mechanism, and also most important first question, most likely to figure out what is the binding partner of this small molecule. The partner can be protein or it can be different macromolecules, but anyway, the target is very important to understand the mechanism of this small molecule. That's why we call this approach as target identification. Usually this target identification step is one of the most challenging part of chemical genetics and also drug discovery. So this week we are going to take opposite approach. I'm going to show you one brief summary of the movie about target identification, and then we are going to move into details of those lectures.
More than 100 years ago, a German doctor by the name of Paul Ehrlich advanced the idea that a chemical substance could selectively affect physiological processes in the body. This idea, which he called the magic bullet, is the basis of modern pharmacology. Practically every medicine in use today is interacting with a protein target and either boosting or inhibiting its function, thus achieving its therapeutic effect. It is estimated that there are more than 2 million different proteins in the human body. We know the function of only a small fraction of them. How can we find out which proteins are deregulated in a disease state and which we need to influence to achieve a cure? Proteins come from genes, and it is somewhat easier to study genes than to study proteins. One approach to finding new drug targets involves comparing the genes of healthy individuals with those of people with a disease. The differences between the two genetic maps can help generate hypotheses on which proteins, or lack thereof, cause the disease. It is also possible to do the opposite. Change one gene at a time in cells or simple organisms and observe the resulting effect, the so-called phenotype of the mutation. If the phenotype has some similarity with a disease state, it can give ideas about the possible relation between the mutated gene and the disease. Yet another, different approach is to start already with a bioactive substance, for example a natural medicine used in traditional medicine, a compound from basic research, or known drugs with unexpected effects. A famous example of this is aspirin, the active component of the willow bark extracts used to cure fever long before modern medicine. Its target protein, cyclooxygenase, is also the target of a number of modern anti-inflammatory and analgesic drugs. It is not easy to find out which proteins bind to a chemical substance. A rich discipline called chemoproteomics offers a wide range of methods to study just that. At Roche pRed, we consistently look for new targets with many different approaches, from phenotypic screening to gene association studies, to chemoproteomics, transgenic organisms, imaging, biomarkers, and many other methods, looking for the gold nugget of information hidden in large amounts of biological data. Identifying the right target is only the first stage of a very long process. But, as a wise man said, a journey of 1,000 miles begins with the first step. And with a potential cure at the end of the path, we confidently begin the march.
How was the movie? Did you get a good idea about the target identification? In target ID, usually we use this small molecule as bait, and then find out the partner from the cells. And inside the cell, there are potentially thousands of tens of thousands of different proteins existing. That's why usually the first step of target ID is to break up the cell, and then make mixtures of protein sets. That process we call cell extract preparation. To make the cell extract, usually we add detergent to break up this cell membrane and then take out those proteins, make it into one mixture of soup. And most common technique in target ID is affinity matrix. In this approach, this is your small molecule. And utilizing this as a bait, we attach fishing pole. And then here is our cell extract. So this is mixtures of different protein. And then utilizing this fishing pole, we do physical fishing experiment. And then only this target protein may bind to this bait, and all other proteins can be washed out. After we purify this target protein, and then we can analyze the structure of the protein by mass spectrometry. Affinity matrix technique also can be called pull-down experiment. Utilizing chemical and take out the old protein sets. That's why this technique also can be said chemoproteomics. To get successful result from affinity matrix, the criteria is the target protein should have quite a high affinity to the molecule. If it is a weak binder, then during the washing step, those proteins can be easily washed out. Second criteria is the target protein should exist in reasonably high copy numbers. If the target protein number is too low, even though affinity is very high, maybe the end result of bound protein amount is too small, and it will be very difficult to analyze that protein. And relatively quite a large quantity of sample is required. And we cannot apply too stringent, too harsh the washing conditions. If you do, then we may lose most of those bound proteins. And weak binding proteins, anyway, will be lost even though we try quite mild washing conditions. So we have a chance only to find reasonably high binding at the affinity at the protein sets. After we isolate out those proteins, we need to analyze them by SDS gel. So that's why too big protein may not move much at all, and too small protein may pass through. So that's why too big or too small proteins is difficult to detect from SDS gel experiment. And the fishing pole is a polymer material. That's why there's always a chance of a non-specific binding of proteins to the solid support material itself rather than to the small molecule. During this affinity matrix procedure, a lot of detergent should be added, and also lots of dilution can happen. So that means if only the complex protein structure has a binding pocket for the small molecule, this dilution and also detergent can destroy that complex, and the binding site can disappear. In that case, we lose the chance to fish out this target protein, especially in complex forms. Also, this affinity matrix technique requires serious amount of SAR study. Okay, SAR is structure, activity, relationships. Let's say this molecule is an interesting compound, and we wanted to find its target protein. Then first step is, we may modify this molecule by adding different linkers. With the linker, we can attach this molecule onto solid support. And if we modify these positions, let's say the molecule loses its activity, and it cannot be utilized for affinity matrix. But in this case, if we modify this position, then still the molecule has activity, then most likely the situation is there is a binding protein, and then they bind very tightly to this molecule around this area. That's why if we modify any of this position, this molecule cannot bind anymore to this target protein, we can assume. Maybe this position is open to the outside, and then even though we add a linker molecule here, then still the molecule can bind to the target protein. That's why it can keep some activities. So this study is called as a structure-activity relationships. In this case, the structure means the molecule's structure, modify the structure, and then activity against the phenotype or the target protein. So we don't have the information about the target protein. Only we can measure is by changing the structure of this molecule and measure its activity. That relationship is called as SAL. Sounds like a very systematic study, but in practice, it's almost trial and error type of approach. By changing different structure of this molecule, we figure out which position we can modify without losing most of those activities. That is SAR. Anyway, after you are done successfully with this SAR study, the next step is you can add this linker and then attach this whole derivatives onto solid support. Then this whole molecule can be used as your fishing pole. This solid support material is usually hydrophilic polymers, usually made of carbohydrates such as dextran, and size of this polymer bead is 100 or the 200 micrometer size, so it's quite big. After you incubate this molecule with your target proteins or the cell extract, and then you can filter off and you can capture only bound proteins to this. So again, to make this affinity matrix, you need to do a lot of SAR study, which is not trivial. Or even sometimes this SAR study does not allow you any position you can modify, because this small molecule is very small and protein is binding to this small molecule, and they just wrap up the whole structure without leaving any exposed space to the solvent. Then you don't have any chance to find reasonable the linker attached to the molecule. But before you try with the SAL, you cannot figure out because you don't have any idea about the binding mode or about the target proteins. That's why SAL takes lots of efforts, and also it does not guarantee your success to make out the good affinity matrix materials. Because of these technical difficulties, you may think it would be much better if there is any other way which does not require you any SAR study. There are some possibilities. If the molecule's binding affinity to the target protein is very strong, and even after you run the SDS gel, SDS is a detergent, and most likely SDS can dissociate the complex of a small molecule to protein, but if the binding is very strong, mild treatment SDS, and the complex can survive this gel running, and in that case, you can label this your molecule with radioactive motif, such as tritium or carbon-14, and then this whole complex can be visualized after gel running. In this case, now maybe it's possible that which protein has a binding to this molecule you can visualize, and direct isolation may be possible. But this approach is only possible when this molecule's affinity to target protein is very strong. Only exceptional case we can apply this technique. But in most of the cases, the molecule's affinity to the target protein is not so high. Now we need another techniques. Let's say this is your active molecule. Then you can look around this molecule structure to figure out which position is to modify easily. Can you figure out? How about this acetyl group? This can be easily acylated on this amine group. Maybe this position may be easy to modify. Then you can think different options. One option can be you can attach one chloride here. Then this chloride molecule, chloroacetyl motif, this is reactive to nucleophile. This is a good electrophile. So in this case, we can call this molecule is good chemical affinity molecules. That means If target protein can bind to this molecule, maybe target protein can bind to this molecule too and may make covalent binding to the target protein. Once you make a covalent binding, then the protein complex to this molecule can survive in SDS gel running. Another option can be maybe this kind of a photoaffinity molecule. Photoaffinity means if you irradiate light here and it can generate very reactive carbene structure, it can make covalent binding to the target proteins. Let's look at a little more details. In case of a chemical affinity, if you mix this together with the proteins, and let's say this compound is binding to the target protein, and then nearby nucleophile, most likely this is a carbon chloride, that's why thiol may be good nucleophile for that, and then it can make covalent binding to this molecule. Then in this case, this molecule to protein complex is very stable. You can catch this whole complex in the SDS gel. This is one approach. Different type of electrophile is also possible to use. Let's say this is a small molecule which binds to target protein, and then which is connected to this epoxide. Then this binding induced a very close location of this molecule, and nearby nucleophile can attack this epoxide, make covalent binding. After that, there is a possibility that you can remove this part and then attach probe to this motif. Then it's possible utilizing this probe to visualize that protein or isolate out this protein very easily. But in this case, this is a stepwise reaction. First step was making covalent binding and exchange this motif here with probe molecule. That's why this is a two-step. This method is developed by Itaru Hamachi, and then his group actively developed this technique further to make it simpler and easier to use. In their next step, they modified this idea by utilizing this ligand and also utilizing this Tosyl group. In this approach, this ligand binds to the target protein, and the nucleophile attack this site, then whole of this group can be removed. Then this labeling with this probe or tag can be achieved in one step. So this is much more advanced the technique comparing to other techniques. Another interesting idea is also developed by the same scientist. In this case, ligand is attached to catalyst, and then active ester molecule is added together. Then this binding of this ligand to target protein can activate the reaction of this active ester, and nucleophile can attack here. Then this protein is labeled with a probe. It's the same as before, but previously those structure of ligand or the complex is destroyed too. But in this example, this original ligand and catalyst part, this is still intact. It's the same with the original structure. That means this structure can be released from this protein and can activate another target proteins. That means this molecule can be recycled as catalyst, can potentially induce or increase those efficiency of this labeling method. In comparison to the chemical affinity, how about the photoaffinity? Photoaffinity case, if we mix together with the protein sets and then irradiate with the light, then this motif can be activated, generating active carbene structure. And this carbene can attack the proteins. And this carbene is so reactive, it is not reacting with a specific other motif of the protein. It can make covalent binding to different position of those proteins. That's why the advantage of this photoaffinity is the labeling efficiency can be potentially higher than chemical affinity. Chemical affinity case, the orientation and also binding site of this molecule to the target protein should be ideally located to nearby to the nucleophile. But in photoaffinity case, it doesn't matter. Any position it binds to the protein, anyway, the activated carbene can attack various positions of those proteins. So the covalent binding formation efficiency can be very high, but disadvantage is it may generate mixtures of different binding product. So it has some advantages and also some difficulties together. If you summarize this approach, let's say this is your drug candidate or small molecule which bind to target protein. And sometimes you can introduce reactivity functional group. It can be chemical affinity or it can be photoaffinity. Let's assume you bind these two motifs to the target protein. Now next step is somehow the tagging part or probe part we need to have here. It can be visualizing the complex or help out to pull out the target proteins. In that case, there's one interesting and very common tools. This is the structure of biotin, and biotin is one of the vitamins, and biotin is a vitamin B7. And the unique and interesting property of biotin is it has very high affinity to the protein avidin. And for research, streptavidin, one kind of avidin, is most popular proteins which we use. And the binding affinity of streptavidin to biotin is very high. The Kd value of this complex is 10 to the minus 15 molar. 10 to the minus 15 is a femtomolar concentration, which is much higher affinity than antigen antibody binding, and almost practically can be considered as covalent binding. Then we'll see how we can utilize this biotin molecule for sorting function. Let's say your molecule is this. Small molecule can be attached to this biotin molecule, and this side is binding motif, and this side, this carboxylate, can be utilized for connection, which is very convenient to use. Then once this biotin is attached to this active molecule, now this itself is a similar structure to affinity matrix, isn't it? If we introduce reactive motif together, now we have three components. One is your small molecule, original active small molecule, and either chemical affinity or photoaffinity and biotin. Then, utilizing this molecule, we can mix together with the protein mixtures. Then, this active motif can find its binding other partner, and then either photoaffinity or chemical affinity, it can make a covalent binding. After that, we can add streptavidin bead. Then this streptavidin can recognize biotin. Then this whole complex can be filtered off. All other soluble proteins can be washed out. This process is very similar to affinity matrix. But the big difference is, in the beginning, we add a small molecule. And then, only at the last step, we add streptavidin bead. Then we reduce those interaction time of bead to the protein mixtures. Then we can reduce, potentially, the non-specific binding to this bead. Once we get this protein, now we can isolate out that protein and then analyze by mass spec. Once we start this target ID process, there may be different scenarios. Let's say this molecule is your active molecule. Then there may be some target binding site and non-binding site. Then utilizing two different binding sites, you can make two different kinds of affinity molecules. One may be you can add this linker to the active binding site here, or maybe in this case you can add this linker the other side. Ideally, your molecule should not bother the binding of your target protein. So these target proteins can bind to the right position. Still, there is a chance that one protein may be responsible for that given phenotype, even the other protein, which by chance it can bind to this motif, but it does not have direct effect to that expected the phenotype. That's also possible. Another scenario is if you modify your active binding site here, then the binding of proteins can happen only to non-relevant binding site here. These proteins, even though they bind to this affinity matrix molecule, but they are not real target. They are called as off targets. Sometimes these off-target proteins can induce a side effect, or it can make it difficult to understand those mechanisms. That's why figuring out the real target from off-target is quite important. Let's summarize the affinity matrix approach. This was your original active molecule. Through SAR, you found this linker position and then attached that onto solid phase matrix. Because of the size of those solid phase matrix bead, which is 100 to 200 micrometer size, this affinity matrix can be utilized only for cell extract to purify your target proteins. In comparison, if you prepare these affinity molecules with biotin and chemical affinity like this molecule is a little bigger than original molecule, but still this molecule has quite good chance to get into the live cells. In live cell, comparing to cell extract, this is much more ordered the structure, and maybe if your target is protein complex, still they have intact protein complex rather than all dissociated the proteins in cell extract. Now, this molecule can go inside the cell, bind to the target protein, make covalent binding. Then, after that, you can make extract. Then already your target protein is bound here. Then you can utilize this biotin and streptavidin column. You can isolate out that protein. Maybe this approach can give you a much cleaner and also more relevant target protein comparing to solid phase affinity matrix. However, both of those approaches still require SAR study. That means after you find your active compound, you need to make many derivatives to figure out where you can attach this linker part, which is still limitations of this approach. To avoid this highly laborious SAR study, there is another strategy introduced. When you send in those small molecules into biological system, how about we send those biological molecules with the linker together from the beginning? And then later we can take out the target protein utilizing the linker. So what does that mean? Let's look at the example. So in this screening utilizing Zebrafish, now we can do the screening with the molecules, but these molecules carries those linker from the beginning. And then if this molecule shows some phenotypic changes, in this case, proper brain development was suppressed. So maybe this molecule is involved in brain development function. Now, this molecule carries this linker, then that's why maybe we can directly make affinity matrix molecules without SAR. How about it? In this case, we can assume this part is only active motif and this linker part is not participating in the binding to the target proteins. If this approach works successfully, then we can avoid those long time-taking SAR study. Then let's practice a little for this affinity matrix in a real example. Now, if this is a whole cell extract, you make a cell extract and run the gel, usually you can see hundreds to thousands of bands in SDS the gel. Now, utilizing this affinity matrix, if you apply this whole protein here and wash out, and only bound proteins you can take out by SDS boiling, then you can see some limited numbers of proteins. Now you can see several proteins here. How do you know which protein is selectively binding to this motif and which protein is maybe binding to the surface of this solid support? That kind of solid support binding proteins we call non-specific binding proteins. To figure out which protein is a selective binding to this molecule, one trick is you can add active molecule. So in this case, now you do affinity matrix the work again, but not only this affinity matrix, you add this active molecule as a competitor. Then the proteins, the target proteins have a chance to either bind to this motif or bind to this motif. If the protein binds to this motif, the affinity matrix bound protein amount will be decreased, and that gel is like this. So comparing to the first one, this band and this band disappeared. What does that mean? Now we have proteins A, B, C, and which one is real target protein to this molecule? Is that A or B and C? Yes, it should be B and C because the target proteins, they bind to this active motif and they have less chance to bind to this affinity matrix. That's why they disappear in this case. To further confirm whether this effect is selective or not, so we can add another molecule. So this is active molecule, this is inactive derivatives, which has the only difference in these methoxy positions. Active molecule has its own meta position. This methoxy is in own para position. Molecular weight is the same. That's why the relationships of these two molecules is isomer. But this molecule is inactive. Now instead of this active molecule, we add this inactive molecule to this affinity matrix and let them compete. The result is like this. This B and C band still bound to this affinity matrix. What does that mean? Binding affinity of a target protein to this motif is very selective and it does not bind to this para position molecule, only it binds to the meta position molecules. Now you can confirm the selectivity of the target protein to this molecule is quite specific even to the small changes of location of methoxy groups. Then you can comfortably say now, at least binding wise, this protein B and C is selective binder to these active molecules. So far we have studied affinity matrix approaches, but still there are different types of strategies. If our targets are really proteins are made by genetic information, then maybe the answer can be found from the gene. Also, maybe the target proteins is very small amount of protein, then utilizing affinity matrix, it may be very difficult to fish out the small proteins. Then we can take different genetic or genomic approaches. One of the possibilities is utilizing 3 hybrid systems. So what can be 3 hybrid systems? So let's review about the transcription. When we studied this transcription the mechanism, we said we need one DNA binding domain and then activation domain bind together to each other, then gene expression can start. As one of those modifications of this system, we studied 2 hybrid systems. In 2 hybrid systems, we can introduce 2 patterns of those protein, and then we can utilize this system to identify one protein's binding partner as another protein. And then also using this gene expression system, we utilize one bait as a protein and then screening out all other proteins to figure out protein-protein interactomes. Now, we further expand this into 3 hybrid system. In 3 hybrid system, we have one protein here, another protein here. There is another small molecules involved here. Let's say this is your active molecules and you wanted to find its binding target. Then in this system, you have one beta system here which has a known binding affinity. Then you can express proteins from cDNA. Then you have target protein candidates library here. Then your screening can be this motif bind to this known partner, and then the other partner as a library, you can screen out which protein bind to your active molecule, and that result can be checked by gene expression. Once you see gene expression, that specific protein is binding to your active molecule, then that is your target protein. So this is a so-called 3 hybrid system. Affinity matrix technique is rely on only chemical affinity to the proteins. That's why it was called as chemoproteomics. But in these three hybrid systems, now we combine some genetic genomic approach together with small molecule affinity and small molecule binding to the target proteins. Another interesting technique for target ID is drug affinity responsive target stability. If you have just a cell extract, if you don't add any molecule, then this molecule, if you add a protease is the enzyme which break a protein. Then they make slice of those protein, and then you can analyze that by mass spec. If you add your active small molecule to this cell lysis, now situation becomes a little different. This active molecule, let's say it binds to target protein. All other proteins can be easily hydrolyzed by proteases, but this bound complex, has a more rigid structure. That's why it is more resistant to protease hydrolysis. That's why they have a better chance to survive. Now, through the mass spec analysis, if you compare this peptide mixture with this, then you can see uniquely different protein which is survived. That can be potentially your target. This technique is also called DART technique. Even before you go for mass spec, you can check that difference from SDS gels. So if you don't add a protease and if you don't add a drug, this is your total protein sets. Now here, if you add the proteases, most of those proteins are degraded, only showing small the peptide fragments here. Now you add your small molecule. Then you have a chance to see some proteins survive better than the other, and this protein potentially can be your target protein. Interesting idea, isn't it? So if you are lucky, without doing any SAR study, by just adding your molecule to the cell extract, you have a chance to find out your target protein utilizing this enhanced stability. Similar concept of ligand-induced stability for the protein has been applied to more sophisticated technique, so-called cellular thermal shift assays. The basic idea is similar. If the protein is bound to the small molecule tightly, it is more stable than the free form. Now that stability is checked by increasing temperature. So, if the protein does not have the small molecule bound, then increasing the temperature, the protein can be denatured, and then it can be precipitated down. If the protein has the small molecule, then forms very rigid conformation, then along the temperature increase of the profile, protein survives much better than free forms, and much slower to denature into the precipitated forms. In real experiment, you prepare two sets of proteins, one without compound, one with a compound, and increase temperature. For each temperature point, you collect those samples and remove precipitated proteins, only collect soluble proteins. Soluble proteins are still non-denatured, they are still stable forms of those proteins. When compared, this is with compound and this is without compound. With the compound, if that protein survived even at higher temperature, that means this compound stabilizes these specific proteins. So this is how you can define, oh, okay, this protein may be the binding target of this compound. So this is the basic principle of cellular thermal shift assays. One potential problem here is, if you want to compare those proteins' stability, you need to select your potential targets. You cannot look at thousands of proteins using this format. That's why you need to have some brief idea what protein can be your potential targets. If you have some candidates among them, you can isolate out those proteins by antibody staining. That staining we call Western blotting. So using Western blotting, you can choose your target protein, potential target protein, and then compare those stability using this compound binding. Let's take an example here. If this is CDK, from lysate, you monitor those CDK stability by Western blotting by adding this compound. If you don't add this compound, CDK2 is slowly denatured over the high temperature. If you add this compound, protein stability is much higher, and then they can survive even much higher temperature, but eventually they denature here. So that's why in this case, this compound may bind to CDK2 quite strongly. Another example, let's just say PARP1 protein, and then you add two compounds here. One is olaparib, the other is iniparib. And one control experiment here is this is control. Now by looking at this example, can you tell between olaparib and iniparib which compound bind strongly to the protein? Can you? Seems like iniparib is almost similar to the control, and only olaparib shows much more stability. So that means olaparib is a stronger binder to this protein, we can say. In this experiment, we fixed one concentration of the compounds and then just changed those temperature. Another format can be fixed those temperature in between and then increase those drug concentrations. So in previous pages, olaparib had a much stronger binding to the target protein, and if you increase this concentration, then the stability of those protein is increased further. But non-binding compound, iniparib, even though you add much higher concentration, still it doesn't increase those stability. In this case, we fixed one temperature. That's why this is called as isothermal dose response, changing of those concentration of the compound and fingerprinting. So this profiling is a similar experiment but different at the dimension. This is called as ITDRF profiling. Here is another ITDRF profiling. Now in this page, we are comparing same cell, but one is in lysate form, one is in intact cells. Protein sets should be similar, but one is destroyed form, one is in intact cells. And utilizing one compound, and then just increasing those concentrations and comparing those stability of those proteins. Which protein is better protected? From lysate or from intact cells? Yeah, it is from intact cells. Even we add a much smaller amount of a compound, the protein can be protected much better than in lysate. So that means inside the cell, the natural environment with a lot of different proteins and macromolecules around, quite crowded the environment. And also maybe the proteins are binding to other partners, making complex. Those status, maybe the proteins are more stable than outside. Or another possibility is, in lysate, the concentration of the compound is just as you add it. But inside the cell, maybe the cell can pump in the compounds. The effective concentration of a compound can be potentially much higher than cell extract. So in these pages, this PD compound has been tested against 4 different CDKs. Out of those 4 CDKs, which proteins are right target of this compound? Seems like a 2 by 2, right? So it seems like CDK4 and 6, they have responding to this compound. These are right target for this compound. And can you tell which protein is more sensitive to this compound? Yes, it should be this red protein, CDK6. Even at lower concentration of the compound, stability increased better. That means sensitivity or susceptibility of the protein is much higher for CDK6 than CDK4. So this is the power of this thermal shift the screening method. If this method can be applied to general target ID, the concept, then can be very, very powerful. Now next question is, how can you expand this to proteomic scale? Rather than only testing several proteins, you may have some idea as potential other candidate.